Right, it's my pleasure to introduce Arun. He's a PhD scholar in the infectious disease program at the Mississippi State University College of Veterinary Medicine and advised by Doctors Larry Hansen and Matt Griffin. Arun earned a bachelor's in fisheries science from Kerala University of Fisheries and Ocean Studies in Kerala, India, and then completed a master's in fisheries science with an emphasis in aquaculture at the Central Institute of Fisheries Education in Mumbai, India. His PhD work is investigating the pathobiology, epidemiology, and phylogeny of alloherpes virus in U.S. farm-raised catfish. So take it away, Arun.
Thank you, Dr. Nick Phelps. It's good to see you, everyone. Today, I'll be talking about alloherpes viruses and some of the work I have done through my PhD work. And so the topic of today's presentation is alloherpes viruses of catfish, latency, virulence, and immunology. Okay. As you all know, the channel catfish industry is the biggest aquaculture industry of the USA, worth an annual sales of 350 to 371 million US dollar. And as of 2020, the hybrid catfish dominates the processed catfish with a 60 to 70% of the production. Today, I'll be talking about the channel catfish virus. So the original channel catfish virus was reported in 1968. And it predominantly affects the fingerlings, and the mortality can go up to 100%. And the typical clinical signs include the exophthalmia, ascites, and the hemorrhage. And the major factor for an outbreak would be environmental stress, particularly the temperature. So I started with this in my PhD work. So there was an initial work carried out in our lab under Dr. Hansen, where they took around 20 to 30 isolates collected over 1987 to 2008 and performed a RFLP analysis. So from the results, you can see there is three distinct groups. So the first group, this one is RFLP A group, this is B group, and there is one isolate which is clearly different from both of these groups. So we would like to investigate further on this. So as a starting point of my PhD program, what I did, I took around 20 to 30 isolates, which we collected from 1987 to 2009, and we did Illumina as well as nanopore genome sequencing. So then we started working on different aspects on these isolates. So the first aspect we were working we were interested what is happening with this isolate, this distinct isolate. And this isolate is came from blue catfish. So it was collected from the Stonewall, Mississippi Delta. The clinical signs were similar to CCV. They do grow in CCO cells. However, DNA polymerase gene identity was less than 98% between the new isolate and the CCV. And as I have already shown, they are distinct in RFLP analysis. So our research question was, is it just another CCV isolate, or is it something new, or is it different? So as a first step in the diagnosis, we did electron microscopy. As you can see, a fully developed herpes virus with the electron dense core, envelope, and a typical icosahedral symmetry. So it does confirm, yes, it's a herpes virus. Second step, we did histology, where you can see some necrosis over a kidney and a congested spleen. Most importantly, herpes virus-induced intranuclear inclusion bodies can be seen in the head kidney. So this further supports our hypothesis. Yes, it's herpes virus. Okay, what's next? In the next step, we try to grow this virus. So here I'm presenting the comparison between CCO cells and Chinook salmon embryo cells. As you can see, CCV produces a typical syncytia where you can see the cytoplasmic spindles and the bridges are formed and the syncytia is formed. And similarly, the blue catfish virus or the virus isolate from blue catfish, that's the same. However, when we look at the CHSE cell at 25 degrees Celsius, only the blue catfish virus grows in CHSE. So that was a differential results we got. So we did, I did another 7 more different cell lines where the CCV can grow in CCO and BB cells. However, the new isolate can grow CCO, BB, as well as CHSE cells. In the next step, we did a serum neutralization assay. So MAB95 is a monoclonal antibody developed by Arkush 1992. He gave us, and we did a neutralization assay. As you can see, CCV neutralization is 1.8, and the blue catfish virus was refractive against the monoclonal antibody. So it says BCHV is antigenically discrete. So this could helps in differential diagnosis. So this is the first point where we got a clear evidence that our new isolate could be something different than the already reported ICHV1. In the next step, we did some challenges. So it's simple. We exposed the channel, hybrid, and blue catfish with the blue catfish virus. And the survivors were kept at 28 degrees Celsius. After a month, they were re-exposed with ATCC type strain. So this is our first challenge results, where the graph is a survival analysis based on Kaplan-Meier estimates. So we used the LC50 dosage of blue catfish virus, and this is the blue catfish, where it registers a 50% survival when they are exposed to LC50 dosage. However, interestingly, as you can see from here, the channel and hybrid registered 100% survival against the blue catfish virus. So it did raise some questions. Do the survivors, that is the channels and hybrids which survived the blue catfish virus exposure, does it give any immunity? So in order to test that, we did the second set of cross protection challenges after 28 days. So to very interestingly, as you can see, the channels and the hybrid catfish, which survived the previous blue catfish virus challenges, registered 100% survival against the CCV. So this is a first report of an alloherpes virus. Previous exposure gives protection against another alloherpes virus. So that was a very interesting results we got. So we did publish both of these results, and the one came just last week as in Journal of Fish Disease. So that's about the blue catfish virus. So we do have enough evidence to suggest that this may be considered as a new species, the blue catfish virus, blue catfish alloherpes virus. And we think that it is sufficiently different from the CCV or ICHV1. In a different aspect, so as I earlier discussed, we have two RFLP group, A and B. We would like to know what is the prevalence of these variants in the hatcheries, in terms of latency. So I started the work with our real-time protocol. However, the protocol was not optimized for the latency work. So as you can see, these are same samples where you can see a cross reaction between ICHV 1A group and 1B group. So there is a cross reaction. So this has led us to the development of new assays. So what we did, we took 4 A group isolates and B group isolates. We did Illumina sequencing and we assembled the genome. From that, we designed separate assays for A and B. Initially, the specificity of the primers were tested with a cyber green assay. Later on, it was tested with the EMM master mix with the SPUD as internal positive control. And we did the validation according to MIQE guidelines. So we use the ATCC type strain as A group standard and LA 87193 isolate as B group standard. As you can see, this is our validation results. We tested against 7 folds of magnitude. So these are 7-fold dilutions of the standards to test the specificity and sensitivity and the linear dynamic range of our assay. So these are our standards and the green ones are my SPUD. So in the presence of SPUD, there is no inhibition. And further, we test, so we are looking for the latent variance. So the problem is we are expecting for a very low copy number. So we would like to use more amount of DNA. However, before doing that, we would like to test it whether, so we tested 1 microgram of DNA in our reaction mixture, and we verified with a spike assay that up to 1 microgram DNA presence does not inhibit our reaction. And we tested this against catfish, CCO, tilapia, and cold DNA. And there was no cross-reactivity with the fish host DNA. So similarly, in our second protocol, which is for B group, we did the validation. As you can see, there is no inhibition here also. And we got an efficiency of 95% with a slope of 3.4. So once our assay is validated, we started collecting the fry samples from commercial hatcheries. So we selected 6 hatcheries. We did 3 sets of sampling with an interval of 10 days. Initially, we collected 200 fries from all the available troughs. So the earlier report of the latency of CCV is 10%. So considering that, I selected 60 individual fry from each sampling point. So we have 3 sampling points, which contributes 180 fry. I did the DNA extraction, individual fry DNA extraction, and I did the PCR. So this is my PCR results, the amplification. The red ones are my standards. These are my positive amplification, and there it is my SPUD. So this is for B group and A group as an example. But we did encounter another problem here. Here, as you can see in the standard curve, this is my standard, the last dilution which shows up after 36. We don't have much samples after that. It's fine. So this is a PCR with a 100% efficiency and 3.3 slope. However, in this case, as you can see, this is the last dilution. Even though it's at 38, there are amplification after the standards. So what we should do about these samples? That was our next question. Because the latency, the genome copy numbers are very slow and they tend to show up after 30 to 33 cycles. So we are proposing a new criteria for the detection. In my case, I have used one individual fry, and I did 3 technical replicates. So out of 3 technical replicate of one individual fry, if the 3 wells shows positive, we call it as positive. If it is 2, suspected positive. If it is 1, suspected negative. Now, for the baseline setting, I have used the Biorad endpoint utility, where it averages the signals of last 5 cycles. So 35 to 40. It just averages the signal, that means RFU, and take the 10% of that as our baseline setting. Then the software does this calling. Later on, I did the data analysis. So let's look at the results. So this is the results for hatchery 1, so prevalence of our variants. So as you can see, 6.6% positive A group and 3.2 B group. And we performed a chi-square post-hoc test on all our data. There is no significant difference between A group and B group. In hatchery 2, as you can see, we could not find any A group, and we find only 3.3% of B group. That is hatchery 2. When we look at the hatchery 3, so we have some more positives, and we could detect both A and B in the 3rd hatchery. Still, it's a low percentage. Again, in hatchery 4, there is a significant difference between RFLP A group and B group in hatchery 4, with a 7.45% of RFLP B group. However, this is getting more interesting, where hatchery 5, as you can see, nearly 80% of our sample is positive, compared to the hatchery 1, which was just 3.3%. So there is a significant difference between A and B too. In hatchery 6, as you can see, almost 97% of the fry, individual fry. So I did the individual fry. So we have a 97% prevalence or positive samples in hatchery 6. And it does have both A and B group, and when we look at the prevalence of these variants across the industry, when the data is combined, you can see like 11.4% of A group and 12.8, which is around a 25% prevalence in the catfish industry, based on just 6 representative hatcheries we selected for our survey. So as I can say that the variation in the prevalence is mainly depends on different hatcheries and their management practices and broodstock management. So as a conclusion, based on host range and immunogenicity evaluations, genome sequence data, the new virus, the blue catfish alloherpes virus, should be given a consideration as a new species of ictalurivirus. Newly developed PCR assays are robust in detection of newly identified ICHV1 variants. Prevalence of each group varies among individual operations. So this work provides a foundation for future epidemiological studies investigating ICHV1 and BCHV in US catfish aquaculture. I would like to thank my committee members for their tremendous support during my work. And I would like to thank my collaborator, our different universities and labs. These are my funding source. I would like to thank ICAR India and USDA and Mississippi State University for their generous funding. These are some of my references. And thank you. So
Great. Thank you very much, Aaron. That's a great presentation. Virtual round of applause to you.
thank you.
Yeah, I don't see any questions in the chat box yet, so feel free to drop them there or unmute yourself and ask Aaron directly. We've got a few minutes for Q&A.
Hey there, this is Isaiah from the Minnesota Department of Natural Resources. I have a question. Great presentation. I'm just kind of wondering what's the what do you think is the sort of the etiology of how this virus is getting spread in the hatchery? Like, is it like vertical transmission from rootstock to fry, or could it potentially be integrated in the genome, or what do you suspect? Because that's pretty interesting that some of these fry populations have such a high prevalence.
Yes, so there is two parts for your question as far as how they transfer. Is it integrated to genome or is other? We do not, I did not perform any experiments on that, and we do not have much data on that. There is a hypothesis, I would say, like they say, it could be through the ovarian fluids. However, we do not perform any experiments on that. The fry, what we are seeing, we can say that they are vertical transmission because directly from the egg, I'm taking the swimmer fry. So they have not been exposed to the pond yet. So and my samples consist of both channel and hybrid. So hybrids are, the preparation happens inside the hatchery and they directly goes to different troughs. So I have even data based on troughs. So they are transmitted, the transmission is vertical transmission, but what is the mechanism? I'm not sure. And does they do the horizontal transmission within the trough? I don't know, because we did not perform, because the reason is we do not. So when we look at the amplification curves, the curves are usually showing up 33 and above. So which is an indication of a latent infection. But if that comes at 30 or below that, we can say that it's an active infection, and it really depends on the hatchery conditions and management and are the fries or stress. So is it possible? Yes, and there have been outbreaks in the past reported in the inside the hatchery too.
Okay, thank you. And just one follow-up question too. Did any of your I know that for some alloherpes viridae, they're having a circularized genome is a good indication of actual latent virus, or you can sometimes find latency associated transcripts. Are there any of those that is that true for channel catfish virus 1? And have you looked into that at all for this new blue catfish virus?
No, we did not look into that aspect. So it could be circularized, I guess, but we do not have a, I did not perform any experiment on that. So
yeah, that would be interesting to know whether exactly. There's some evidence of kind of like biological latency, because you know that term gets thrown around a lot, and it's hard to sometimes pin down exactly you know, if this is like true latency or if it's just sort of non-replicating persistent virus. That's something I deal with koi herpes virus. So I'm curious about that with other alloherpes viridae. So,
but one thing I can confirm is out of the one hatchery where we did the survey, they had an outbreak later.
Okay.
So that's only confirmed because, but whenever we try to do the cell culture of these fries, they don't grow. So they don't. Yeah. They're not actively replicating, I would say. However, only thing happened is like later the same farm reported an outbreak in the ponds. So what we are detecting, I would say they are not in active reproduction state. They are still in latent. So because we cannot grow them in the cell culture. That's the only limited data we have right now.
Okay. Well, thank you. Great work.
Yeah. Thank you.
All right. Esteban, I see your hand up. Do you have a quick question?
Well, they weren't really quick, Nick, but great talk, Arun. I had a couple of questions, and I don't know if you have time to answer them or not, but the first one was there were some pretty high prevalences, in some of your sampling, populations, and I'm just curious if any of them actually were reported by the farmers as outbreaks. Like, did you have actually clinical signs of disease and dead fry, or if it was just like a regular surveillance for your research, and you just happen to diagnose a pretty high percentage of them. So that is the first one. The second one is regarding the blue herpes virus. That you mentioned that it grew in the
CHS.
I think it was that. Did you say Chinook salmon, the CHSEs?
Yeah.
So those one you incubate at lower temperature, right? Around 20 degrees Celsius typically. I don't know what temperature you incubate them this time, but the challenge that you perform in the channels, the blue and the hybrids, was at a pretty high temperature that is similar to where the outbreaks of the Ictalurivirus 1 occur. And I'm just curious if you have thought about challenge them, like those fish species at lower temperatures, as this virus seems to be able to still cause cytopathic effect in cell lines at a pretty low temperature. So it might be temperature and not really host specificity. So those were my two very fast questions.
Okay, so let me do two quick replies. The first one, the hatcheries which had the high the viral load, they did so. The one which had the highest one, the last 5 and 6, they did not report any cases to the diagnostics. And nowadays, farmers usually doesn't report the CC, seeing that we don't have any antibiotics or antiviral agents for that. That could be one reason, but only I can confirm is that within that there was another hatchery which I did the surveillance. They reported the case to the diagnosis, but the one where the high prevalence where they did not report any case to the diagnosis. First thing. Now the second question, as far as the CHSE, the growth in CHSE, so that's pretty interesting, because if we do it at 23 degrees Celsius or 24 degrees Celsius, no CPA. It happens only at 25. And if I try to raise the temperature about 25, that the cells become unhappy. So it's really a balance regarding the cell line the growth. So the blue catfish virus does show a CPE at 25, and in that temperature, the CHSE cells were okay, or I couldn't find anything weird going on. But above that, yes, CHSE cells become stressed, and below that, virus doesn't grow either. So it's a pretty small area. And the third question was about the challenges. Yeah, we did all the challenges at 29.5 degree. We used the heated water. We bought using a boiler. We heated and distributed to all the tanks. So all the challenges, the CCV challenges as well as the blue catfish virus challenges, were done in 29.5. And I can say, yeah, there is a good correlation between the temperature and the onset of disease too.
All right. Well, thanks again, Aaron. Excellent presentation. And you have one question in the chat box. If you could jump in there and answer it quick. But we're going to